Hello everyone, my name is Farshid and I'm the Serology Chief Scientist at Dorevitch in Melbourne. I started working with Oz Diagnostic Hiplexes at Pathology and later on extended my collaboration with Oz Diagnostics at Tertiary Pathology in Darwin. We increased the capacity of the PCR testing at Royal Darwin Pathology from 30 tests a day to more than 4,500 tests a day. We're one of the main users of Oz Diagnostics UP3 systems in Australia. Our lab was doing the majority of the PCR tests for the repatriation flights. We were among the first laboratories provided COVID testing in Australia, including the samples from the passengers landed on the Christmas Island and the passengers of the Diamond Princess cruise. We were never short of the supplies for COVID testing as we were using a different platform for extraction and we were also using the Oz Diagnostic kits for COVID. This was the advantage of using an Australian company, which was very supportive and we were always prioritized. On top of all of these, the customization and improvement of the detection of the new variants of concern was quicker than other well-known bigger companies. Apart from COVID, we were also doing the beta testing for Oz Diagnostic other two multiplex assays, the Mosquito-Borne panel and the parasitic panel. The Oz Diagnostics Mosquito-Borne panel includes the following targets: dengue, parvovirus, Ross River, chikungunya, Zika, Murray Valley encephalitis, Japanese encephalitis, West Nile, Kunjin, malaria, and antimalarial drug resistance. The patient was a 46-year-old Aboriginal female from Tiwi Island. She was hospitalized in 2021 with symptoms indicative of MVE. Both CSF and the whole blood were PCR negative for MVE and JE virus. The patient deceased and the tissue specimens from different parts of her brain and spinal cord were sent for PCR testing. Interestingly, right thalamus and right cerebellum were positive for both JE virus and MVE on the Oz Diagnostic Mosquito-Borne panel, which was going through the beta testing at the tertiary pathology at the time. In this slide, you can see that the right thalamus and the right cerebellum were the only tissues which were positive for both JEV and MVE targets. And the rest of the samples sent for PCR testing were all negative. Also, you can realize that the JE takeoffs are earlier than MVE takeoffs, which is showing a little bit higher concentration of JE targets. The takeoff of the JE target in the right thalamus specimen was 23.57. However, the takeoff of the MVE target was 24.26. Similarly, in the right cerebellum tissue, the JE target takeoff was 24.19. And the takeoff of the MVE target was 24.42. However, the chances of these samples being positive for both targets was very low. This could be due to false positivity of one of the targets because of the non-specific amplification. Interestingly, back in 2020, in one of the alphavirus and flavivirus RCPA-QAP modules, sample MAF 2007 was also positive for JE and MVE targets at the same time, and the JE target was stronger than the MVE target. The survey report revealed that this sample supposed to be only positive for Japanese encephalitis virus and not the MVE, and reported a discordant result for the MVE. These findings were transferred to the Oz Diagnostics team for review along with the extract samples and the amplification products from the PCR run. The Oz Diagnostics R&D team started to redesign both assays as they found the MVE was crosstalking to JE target. The new redesigned product showed no positivity for the MVE target and was only positive for JE target on the right thalamus. However, in the right cerebellum, due to the deterioration of the genetic material and the low concentration of these targets, we only had a very late amplification for the JE target, which wasn't called positive. Still, we could see the melting curve of the late amplified product. Both right thalamus and right cerebellum specimens were negative for MVE on the new redesigned assay. The RCPA-QAP MAF 2007 sample was also reran on the new assay and only showed positivity for the JE virus target. As you can see, there was no sign of positivity for MVE. The sequencing result analysis done through Oz Diagnostics on both amplified products from our run, which is run 1383, and the Oz Diagnostics in-house run, which is run number 121, confirmed the target match for JE rather than MVE. Please note the lower E value in these results showing the higher similarity between the sequences. The referral lab results from CSIRO, Pathways, and the FSS confirmed the JE target in the right thalamus and the right cerebellum specimens. Vitral assay was not as sensitive as the other referral labs assays. CSIRO was able to obtain a complete JEV genome sequence which was clustered within JEV genotype 4. This cluster was from Indonesia and Vietnam. As there was no previous local infection of Japanese encephalitis virus in Northern Territory, the question is, where is the source of this infection? The answer can be found in the life cycle of this virus. Water birds are the maintenance hosts of this virus, and with the change in the weather patterns, they might have flown to new territories. Migration of these birds to TV Island could have transferred it to the Culex mosquitoes in the region, and from the mosquitoes to humans. This means there is a higher chance of seeing of these infections at the top end in the future due to the weather changes and consequently the change in the water bird migration destinations. At the end, I wanted to thank Dr. Rob Bird, the clinical microbiologist at Territory Pathology, Malik Saka, the medical scientist at Territory Pathology, Dimitri Kuefta and Nathaniel Tan, and the R&D team from Oz Diagnostics. Thank you.